Dr. Huduba, so good to see you.
Dr. B, welcome to The Point at Otterbein University. Let's take a walk.
You got it, you got it. So listen, so you're an engineer and you build devices at the nano scale, but you use DNA technology. Tell us about that and tell us about this thing called DNA origami.
When people think of, oh, you do DNA nanotechnology, they always think of genes and things like that, right? The stuff that make you, you, and me. But to us, in DNA origami, DNA is our brick and mortar. It's our steel, it's our cement. It is literally the material that we make machines out of. The other good thing about DNA is it actually assembles itself. There are 4 what we call nucleotides to DNA, A, T, G, and C. And the rule is simple. A binds with T, C binds with G. And so it's almost like a puzzle piece. What we do is we provide the pieces and provide the blueprint of what we want the puzzle to look like. But then if we mix all of those puzzle pieces together, put it under the right conditions like temperature and salt, the puzzle will actually form itself. The scaffold is going to fold into our predetermined shape, and that's why we call it DNA origami.
Are there therapeutic uses or applications for this?
Absolutely. So colleagues of mine at The Ohio State University have developed a structure that they call the horse. It's called a horse because of the Trojan horse, right? The story of the Trojan horse. So a lot of times cancerous cells know when we're trying to give it cancerous drugs. And so before those drugs can get to the nucleus and do their dirty work, they're actually spit out from the cell membrane. So what they've done is actually hidden the cancerous drug inside of DNA origami nanomachines. So they're absorbed by the cell, absorbed by the nucleus, they disintegrate in the nucleus and release their payload.
That is so cool.
So that's not even the most exciting part. Dr. B, I got a surprise for you.
What?
Right now, I have us set up in the lab, so you get to go and make your very own DNA origami Trojan horse.
Are you kidding me?
I am not.
That's awesome. Let's do this.
All right. All right, so we're here. We're gonna make some DNA origami. We're gonna make a nanostructure called the horse. You're gonna mix all of our ingredients and put them in our water bath so we can fold this nanostructure. DNA origami consists of 2 primary ingredients, the DNA scaffold and the DNA staples. This first ingredient you're adding is what we call the scaffold. So the scaffold is the major foundation of the structure. Think of it as the scaffolding of this building. All right, the next ingredient is called the folding reaction buffer. All right, this third ingredient is the salt. Our fourth ingredient here, we call this the working stock. So this is all of the other DNA staples. There's hundreds and hundreds of staples in there that are going to anneal with the scaffold to ultimately make the structure. So that's when you have your 2 single-stranded pieces of DNA separately. Annealing is when they come together to make that double helix.
Got it. It's kind of like 2 pieces of Velcro coming together.
Exactly. So let's go ahead and mix this up. All right, and so now what we want to do is we're going to put it in our water bath between 65 and 70 degrees. For 5 minutes, we're going to melt it at a really high temperature. And then what's going to happen is we're going to put it in what we call the folding temperature, which we've experimentally determined to be 52.5 degrees. Okay. We're going to put it in there for 10 minutes, and that's when all of these pieces are going to very nicely anneal, but fold together, again, origami, and make our nanostructure. All right, so now we're going to load our structures into the gels. What we're going to do is basically put what you made into these little pockets in this jelly-like thing that we've created. Right? And DNA is negatively charged, so we're going to apply a voltage across here, and what's going to happen is that negatively charged DNA is going to go towards the positive end of that voltage, and we're going to be able to see it as it travels through. So what this does for us, things that are nice and compact, like our well-folded origami structure, are going to travel faster and all the same speeds. So we're actually going to not only see our structures travel through, but this is also purifying it, because anything that didn't fold well is going to be caught in the web, and everything that's nice and tightly packed is going to streamline its way through. That green light tells us that we're now applying our voltage, and now we wait 40 minutes.
All right, you know I'm nervous.
All right. So what I'm going to let you do the honors. What we should be looking for here is our right 4 bands. Do we see some nice, distinct bands of light?
Oh, yes. Yes.
You have made a DNA origami Trojan horse. You have made a machine out of DNA.
That is truly amazing. So not only are you a great teacher, I heard that you have a Guinness Book World Record.
I do. I was giving a presentation in Germany, and at the end I put up a picture of the Script Ohio that the Ohio State Marching Band does, and asked if there was any questions. And one of the questions I got was, why don't you make that out of DNA? And it was a brilliant idea, and almost 3 years later, I created one, it worked. And it's the smallest logo made with an uninterrupted strand of DNA in the history of mankind.
What is the future of nanotechnology and these devices?
Yeah, I think the best answer to that question is I don't know. That's how science works. Nanotechnology was created in the early 80s. DNA origami was in 2006. We're creating the basic tools, the basic building blocks. But one thing I am really excited about with DNA nanotechnology is actually digital data storage. So just like computer code is zeros and ones, DNA is A's, C's, G's, and T's. It's just a sequence, but it has the ability to store information at a density of about 1 billion times more than what we can do today, and it doesn't need electricity or heat like a transistor does.
Dr. Hudubah, thank you so much for spending time with us.
It was my pleasure.